I'm pleased to introduce Lauren Agent. So Associate Professor Lauren Agent is a clinical researcher from the University of Melbourne and the Centre for Eye Research Australia. She leads a wonderful team working on natural history of inherited retinal disease, and that's called the Venture Study, and is an investigator on several clinical trials, including gene therapy and drugs for different inherited retinal diseases. So Lauren has previously worked on the Bionic eye research program as well in Australia and in the USA at Harvard and Cornell Universities, so very prestigious. And today she'll be providing an update on some of the very exciting technologies that are being developed for people with inherited retinal diseases. Thank you, Lauren.
Thank you so much, Eden. It really is a pleasure to be here today to share with you all a little bit of the work that's going on in Australia. Excuse me. And I really wanted to start by thanking John and Alan for a really great introduction. And one of the really exciting things in this space is how well the researchers are working together in Australia and also internationally as well. I was just talking to someone this morning about the fact that the collaboration that we're seeing across the globe is really starting to speed up a lot of treatments and other research. So it's an exciting time. Having said that, I always like to sort of caveat what I'm going to talk with, balancing hope with realistic expectations. So I'm going to talk to you about advanced therapies and some of the things that are coming up in the pipeline. And one of the things often when I talk about this is we often talk about therapies and we say, oh, look, they should be around in about maybe 5 or 10 years. And I did a talk recently where someone put up their hand and said, yeah, I've been hearing that for 50 years. So completely understand, and we do get that. And I think a lot of us have family members with conditions that we're searching for treatments as well. So it is a really challenging thing, but we really are at a period of hope for treatments with inherited disease. Now I've got some slides, they're really just to keep me on track, so if you can't see them, I will talk you through them. What I've got first is a graph that sort of shows where the different emerging treatments are focusing. And the really exciting thing for people that are living with these conditions is that there's different treatments being developed for different stages of inherited retinal disease. So we have some treatments which will be more suitable for people at early stages of disease, and then some that are being developed for people that have basically lost all of their vision. And this is really important because we want to be able to offer people as many options as we can. So I'm going to talk today about gene therapy, and we think that gene therapy will be mostly for people that have still quite early disease and they still have relatively intact retinal cells, and I'll talk a bit about why that's important. I'm going to talk about stem cells, and you'll also hear from Anai a little bit later today, who is a world expert in this area, and also the bionic eye, and they're also called visual prostheses, and a treatment called optogenetics. And when the time we get to talking about devices like the bionic eye, these are really treatments that are being developed for people with end-stage vision loss. So you can see that there's a whole spectrum of treatments that are being developed, and I'm really going to try and break it down into simplistic terms to give you an idea of what's coming up in the pipeline. So let's start with gene therapy. So Alan did an excellent explanation of why gene therapies are emerging for inherited retinal diseases. So if we take a step back, the genes in our cells are basically like the instructions for our cells to tell them what proteins to make so that our eyes can function properly. And what happens in inherited retinal diseases is that we have an error in that genetic information, which means that the proteins are not produced properly. They might not produce the proteins, they might produce too much of that protein, and whatever happens, it means that our eyes don't function properly. And so the idea with gene therapy is, what if we could correct that error? And so what we do is, through experts like Alan and John, we identify what the incorrect error is in that genetic information, and then we try and get a copy of that correct DNA into the eye. And so I have an image up on the screen for those of you who can see, which is showing how we do this. So the idea is that we need to get the information into the eyeball. And the way to do it's actually quite clever, and it's by using, at the moment, a virus. So we all have just lived through COVID pandemic. We're aware of how good viruses are at spreading. And so in this purpose, what we're actually doing is using that for good. So we put a correct version of the gene into the virus, we inject that into the eye, and then the virus spreads amongst the retinal cells and basically infects those cells with the correct genetic information so that the retina can start functioning properly again. So that's the simplest version. Now, where it starts to get tricky is that in some cases, the eye's already making too much of the protein, so we don't want to do that. In other cases, the gene might be too big to fit into that little virus. And as Alan mentioned, there's over 200 different known genes, so we can't make 200 different drugs that do this. But this is what Luxturna does, so the treatment that is available on the market at the moment does this. It replaces the incorrect copy of the gene with a healthy version. What we're now starting to move more into is more complex forms of gene therapy, and it's things like gene editing. And some of you may have heard of a term CRISPR technology. And so CRISPR is basically a technique which allows you to edit the genetic information rather than needing to replace just a chunk of it. We're also able to use gene therapies to moderate and modify some of the downstream components of that pathway, so the intermediate parts of going from DNA to going to a protein. And so again, all of these new forms of gene therapy are really about trying to make it more agnostic to the gene, so that we don't have to have 300 different drugs, but we can have a drug which would treat a larger number of people. So gene therapy is a very exciting space, and it's one that we've heard a lot about because of Luxturna, so because there is a gene therapy on the market. And as John mentioned, there's a number of trials that are currently underway or will be underway shortly in this space. So it's a very exciting space. The trick with gene therapy is you do need to still have relatively high numbers of healthy cells in the retina. So if you're someone who's lost most of your vision, we don't think that gene therapy will be as successful. And the rough numbers at the moment, we think that it need to maybe have around about 2/3 of the retinal cells still intact, even if they're unhealthy, to have gene therapy actually be able to work. So again, this is something that scientists are working on, and as the trials continue, we will be treating people at different stages of disease. The next space I wanted to touch on is pharmaceutical agents, and again, John's done a beautiful introduction to this. In Australia at the moment, we have 2 oral medication trials going. So there's one called Nucuity, and that's an antioxidant treatment which is being trialled for people with Usher syndrome. And the idea with this is that the oral medication basically acts to protect the retina and slow down the progression of the inherited retinal disease. Interestingly, that trial is really focusing just on Usher syndrome. There's another trial happening in the US, which is a similar compound. It's slightly different drug, but that's actually going to be trialled in people that have different forms of retinitis pigmentosa. And again, the idea is it's an antioxidant, it's protecting the retina and hopefully going to slow down progression. The other trial that John mentioned is Tinlarebant, and this is a study that's called the Dragon study. And this particular drug is in patients with Stargardt disease, and it's being tested in adolescents, in younger people. And the idea with this is trying to reduce the amount of vitamin A that's getting to the eye, because that can lead to toxicity in people that have Stargardt disease. And so these are quite exciting trials because, obviously this is an oral medication, it's a tablet that people are taking once a day, and there are obviously more of these being developed in the pipeline as well. So stem cells, again, and we'll talk a little bit more about this later, these are a really exciting area because of the way that technology has changed over the past few decades. So a stem cell is basically a cell that can replicate itself or can differentiate into many different cell types. So a stem cell might become a bone, or it might become a piece of hair or a skin, and this is sort of the beauty with stem cells. They can be what we turn them into. Historically, stem cells were taken from embryos, so usually those that were being used for IVF. And so obviously that had a lot of ethical and moral questions about using those for treatments. So back in 2006, a group in Japan actually developed a technology called induced pluripotent stem cells. It's a bit of a mouthful. But the idea with this technology is you can take a sample from someone. So you can take a piece of their skin or a bit of their hair, turn it back into a stem cell in the lab, and then grow it into what you want it to be. So you can take a skin sample from someone and then make it back into a retinal cell, so a photoreceptor cell or another type of cell in the eye. And that allows scientists to do a whole range of things. So we can then grow up a series of cells in a dish to look at how that person's particular disease is progressing. We can use that to test drugs. So we can say there's this particular problem with the person's cells, let's have a look at how a drug helps. And we can also use it to identify new treatments for that condition. The other thing that's quite exciting with stem cells is we might be able to use them to replace the cells in the eye. And so that has been trialled to date. So you can either put the cells onto sort of a scaffold and implant that into the eye, or you can actually inject some of those cells into the eye as well. And the idea here is that you're replacing the sick or dead cells in the eye with new ones. This is still not really in clinical trials for inherited retinal diseases yet. It's been more used in macular degeneration, but obviously a space to watch. So the last treatment area that I wanted to touch on is really the treatments for the end stages of eye disease. So as I mentioned before, gene therapy and some of these oral medications are really more for people at the earlier stages. When we get to people that have lost quite a lot of vision, the treatments that are being developed are either bionic eyes or treatments that are called optogenetics. So I'll start with bionic eyes. Many of you in the room will have heard about this. This type of research got a lot of press because of how successful the cochlear implant is. And as Eden mentioned, I was lucky enough to be part of the group that worked on the Bionic Eye project. And the idea is that you have a pair of glasses that's got a camera on it, takes a picture of what you're looking at, and it translates that into electrical signals. And you can either send those signals directly to the eye or back to the brain. And so the treatments have been trialled in 7 people in Australia, and their baseline level of vision was what we call bare light perception. So it means if you sort of close your eyes, that's sort of the vision they had at the start. So they could see if the lights were on in a room, but they weren't able to see any people or objects. What we found in the clinical trials of the bionic eye devices is it allowed people then to be able to see large objects, make out things on a table, and probably most importantly, make out where people were in a room. And that was really the thing that people most spoke about. They could then identify if someone was in a room, you know, where the barista was at the coffee counter, those sorts of things. So the bionic eye is a really interesting space because I think there was a lot of expectation that it would be the next cochlea. And the thing to understand is that the eye is much, much more complex than the ear, and my ENT colleagues hate it when I say that. But when you think about the ear, we're basically trying to reproduce the action of about 15,000 hair cells in the cochlea when we're putting a cochlear implant in. In the retina, we're talking millions of cells. And those cells are very unique. So some cells react more to colour, some cells react more to motion. So trying to reproduce that just by putting electricity onto the retinal cells is really challenging. So having said that, work is still underway. So there's been about, I think now we're up to around about 480 people around the world have had bionic eye implants. There have been 3 companies that were started, one in Germany, one in France, and one in the US, that was selling bionic eyes for a time. What happened then was the COVID pandemic, so those companies have unfortunately all folded. But the work continues, and so these devices will still sort of continue to be developed as well. For me, one of the really exciting things is how electronics and devices like this might actually work in conjunction with other drugs or gene therapy or stem cells. So there is work going into, you know, if you give someone a different type of treatment, can you also use electrical stimulation to help the eye absorb that drug more effectively, for example. So I think what we'll see is more and more the electronic engineering world and the pharmaceutical world in IRD will probably come together and hopefully come up with better and better treatments for us. The final option I wanted to talk about briefly is a treatment called Optogenetics. And falling into this broad category is also some drugs that are called molecular photo switches. And you can see we love our big names in science. But basically the idea behind these different drugs is that in inherited retinal diseases, the photoreceptor cells die. So the cells that react to light are the ones that mainly get sick and die. The connecting nerves are relatively intact. And so the idea is if we can actually change some of the cells that are still remaining in the retina and make them responsive to light, we can kind of take over the function of the photoreceptor cells that have died. And John mentioned before the Kiora study, which will hopefully start in Australia soon. So that's sort of the premise behind that. And there are other trials going on overseas as well. So I wanted to sort of finish up there, just I guess with that high-level overview of the different treatment options. As I mentioned at the start, it is a really exciting time. From when I started working in inherited retinal diseases 15 years ago, there is so much more opportunities now than there were back then. However, obviously there's still challenges, but we've got really, really amazing scientists and clinicians working on these options. And I think one of the things that I'm really seeing too is we've got great support from patient support organisations such as Retina Australia, and obviously the work that's Eden and the team are doing here. And so it's really about communicating what we're doing and having open discussion about what these treatments might mean for everyone in the future. So thank you for listening.